Every now and then, you get some bit of biology news coming out of a lab somewhere, about some arcane contorted conglomeration of cells that they've grown in a petri dish, or some kind of purified biochemical extracted from a poisonous beast halfway across the world. The news is often shocking and incredible, and most of all, it's weird. In my opinion, this is where you find some of the most exciting stuff in the field. It's kind of like the engineering breakthroughs in laboratory environments. The engineering breakthrough is usually incredible, and if it can be scaled up, the implications for all of human civilization are groundbreaking and unimaginable. Except, in virtually every case, it isn't feasible or economical to scale up, and thus these engineering breakthroughs are notorious for never getting out of the lab, as they say. Except, when you get enough research done, and enough of these breakthroughs happen in a particular subfield of engineering to sufficiently advance a particular field of knowledge, you do eventually get a viable technology that humanity can enjoy. The news that I want to tell you about is kind of like this, but it's biology instead of engineering. Several years ago, biomedical research began to learn how to grow small organ structures called organoids. It began with simple organs, like the bladder. This particular research on creating artificial bladders would, after a series of breakthroughs, come to develop viable human-sized bladders that are intended for organ transplant. And that's a real medical achievement that came about after years of this mind-blowing but lab-limited work. Other organoids that have been developed to one degree or another include hearts, lungs, livers, kidneys, even though kidneys are a little more difficult than normal because of the cellular structure that has to be replicated, but we've even grown organoid brains. And it's these organoid brains that I want to talk to you about today. This avenue of research began by producing small globules of neurons, which were arranged in simple patterns to create these really primitive neural networks. Over time, scientists learned how to grow larger and larger brain organoids, with increasingly complex patterns that even began to exhibit similar activity to real living animal brains. Now, the news that I want to tell you about today is the next event in this process of development and breakthrough. In this experiment, published in the journal Nature Neuroscience, the researchers began with human stem cells, which they differentiated into various types of neural cells, which they then grew into neural tissue, which they then grew into a complex brain organoid. The organization and structural complexity of this brain organoid was described as being similar to that of a human fetus during weeks 12 to 16 of the pregnancy. Madeline Lancaster was the lead researcher on the project, and when asked about any potential feelings or thoughts or awareness that these brain organoids might have, she said that the brains were far too small and primitive for that. They weren't complex enough to have anything close to a walking, thoughtful experience. Mrs. Lancaster said, quote, "It's still a good idea to have that discussion every time we take it a step further, but we agree generally that we're still very far away from that," unquote. To put the organoid in perspective, it only had a few million neurons, which is on par with a cockroach. A mature human brain, in comparison, has 80 to 90 billion neurons. Okay, so maybe you thought that growing little mini brains in a petri dish was pretty wild, was pretty extreme, on the cutting edge of biomedical science. And you would be right, except the researchers really turn it up a notch. This is where it gets really crazy. These brain organoids are straight out of science fiction, but it's this point that the experiment really takes on its breakthrough nature. So the researchers surgically extracted a 1 millimeter long spinal cord from a mouse embryo, as well as some of the surrounding muscle tissue from the mouse's back. And keep in mind that this was from a live mouse embryo, so this developing spinal cord and developing nervous system would have innervated a lot of this muscle tissue. That'll be important, and you'll see why in just a second. When this length of spinal cord was placed near the brain organoid, the biochemical cues encouraged the growing, pulsing brain tissue to send out grasping neurons to find, connect with, and integrate into the spinal cord. They grew to connect together, and get this, the brain began to send out electrochemical signals called action potentials down the spine along the mouse's nerves to stimulate the muscles and cause the muscles to shiver and twitch and flex. This is wild. It's been described as a big step for the field, although I think the words "a big step" doesn't really capture the magnitude of the weirdness and the medical value here. The weirdness should be obvious, but the medical value to this experiment, to be brief, is twofold. They've developed a technique to grow large quantities of brain organoid tissue, overcoming the challenge of growing veins to keep the tissue mass properly innervated so that it can receive oxygen and nutrients. This technique that they developed allows for the generation of larger brain organoids, and a larger brain organoid is more complicated, and so can be used in more sophisticated and more complicated experiments. The other medical benefit of the research is that now we've seen that cortical tracts can grow and explore beyond the brain to find and innervate other tissues, like the mouse spine and the muscle tissue. This is a giant leap for the medical science of restoring damaged nerves and restoring function to paralyzed tissues. These medical discoveries can help the study of epilepsy, schizophrenia, motor neuron disease, and all manner of other neurodegenerative diseases. This is medical technology straight out of science fiction. In a sense, we are manifesting our imagination into reality, which is creating a better future for everyone.